Good morning, everybody. Ladies and gentlemen, before starting, I would like to ask anyone who is physically participating in this dispensation to switch off any device that might interfere with the defense. And those participants virtually attending the session, I would like you all always to mute the microphone except the external examiner. So my name is Betelet. I'm the faculty at Addis Ababa University. I'm also the deputy director at Africa Center of Excellence for Water Management. So I'm pleased to chair the dispensation of Ms. Muluwork, who is our PhD student. She will be defending her dissertation to earn the degree of Doctor of Philosophy in Water Management, in Water Science and Technology track. Her PhD dissertation is entitled by Biodesalination of brackish water, coupled with biodiesel production using native microalgae species isolated from saline Rift Valley Lake, which is the Lake Azeka, Ethiopia. Her work was supervised by Professor Felleka Zoga. He's a professor of Addis Ababa University, also the director of Africa Center of Excellence for Water Management, and co-supervised by Dr. Debnek Ekple of Addis Ababa University and Professor Ndal Kachos Haile of the Center for Environmental Solutions and Emergency Response in USA. Her work will be evaluated by two evaluators. One will be acting as an external and the other one as an internal examiner. Okay, so Dr. Ashraf Ali Hussein, who joined us today virtually with the capacity of external examiner from United Arab Emirates University, will be acting as an external examiner. So just to say something about Dr. Ashraf, he has over 20 years of experience practicing environmental engineering with an emphasis on biological treatment of environmental pollutants. He is also a licensed professional engineer in Nebraska. He is currently an associate professor at the United Arab Emirates University. He taught earlier at the University of Nebraska, Lincoln, and he served as R&D team leader at BioAir Solutions and wrote air pollution permits for higher profile industries at Nebraska Department of Environmental Quality. He was an ORISE postdoctoral fellow at the US Environmental Protection Agency's National Risk Management Research Laboratory. His research areas encompasses advanced water and wastewater treatment processes and development, and also emission control technologies, biological treatment of recalcitrants, air toxic, use of innovative technology in atmospheric and indoor air pollution control, treatment, fed transport of emerging contaminants. Dr. Ashraf has over 50 peer-reviewed publications, and his works have been cited over 1,608 times. He has been recipient of 2 prestigious Air and Waste Management Association Awards, including the Philistine Memorial Scholarship and the 2011 PhD Dissertation Award. And thank you, Dr. Ashraf, for accepting our invitation. Right?
Thank you.
Our second evaluator, who is our internal evaluator, is Dr. Adey Desta. She is also an associate professor in environmental microbiology and biotechnology in the Department of Microbial, Cellular, and Molecular Biology at Addis Ababa University. She has received her PhD in applied microbiology from Addis Ababa University in 2014. Her research focuses on microbial ecology of biological wastewater treatment systems, focusing on the dynamics of key communities in xenobiotic degradations. Through research fellowship from German Academic Exchange Service and African Bioscience Challenge Fund, she conducted research on detection and monitoring of microbial communities in biological wastewater treatment plants of resource-poor settings. Through the research fellowship from University of Michigan, African Presidential Scholars, she has been involved in the fate of microbes during the process of nutrient recovery from human urine. Since 2016, Dr. Adey has conducted research on monitoring resistome during waste fertilizer processing in Ethiopia through the funds she won from Addis University, Peer USAID, in partnership with and SIDA, in collaboration with the SLU in Uppsala. She has also been involved in One Health research with focus on water and wastewater management in cities of low income countries. She has authored and co-authored 21 publications in the above mentioned areas, and her work has been cited over 760 times. Please allow me to welcome both our examining members to Ms. Muluwork PhD defense. So without any further ado, I would like just to go through the procedure of the defense. So Ms. Muluwork, you will have 45 minutes to present your dissertation, followed by 30 minutes Q&A for each examiner. Right? So an examiner can ask one question and seek for a reply, or you can ask 3 or 4 questions also and seek a reply as well. Then later on, the participants will be invited to ask questions, if any, and finally the floor will be open to the main supervisor to relay their message to the PhD students after announcing the results. So now, without further ado, I would like to give the floor to Professor Fallaka to introduce Ms. Muluwork, then the defense session will initiate. Thank you.
Thank you very much. So I think you have already introduced Muluwork. Muluwork did, I think, she has been here, she has been with us here for the last 5 years. And I think that she has selected a challenging topic, that's biodesalination with microalgae. But with all the challenges, I think she did very good work, and I wish good luck for your defense.
All right, thank you, Professor Fallaka. So, Ms. Muluwork, mic is yours, and good luck to you.
Okay, thank you very much. Hello, everyone, and good afternoon for those who are abroad. My name is Maru. I'm a PhD student at the African Center of Excellence for Water Management from Water Science and Technology stream. Before starting this PhD project presentation, I would like to express my deepest gratitude to the examiners. Thank you for being willing, for your time and effort in evaluating this PhD thesis. I would also like to thank the chairperson, Dr. Batalaita Kolla, and all of you who are attending this presentation, both virtually and in person. My PhD project is on the biodesalination of brackish water, which is coupled with biodiesel production using local microalgal strains isolated from Lake Basaka. This is one of the saline lake found in the Ethiopian Rift Valley. This work is supervised by Professor Fallaka and Dr. Dammaka Kafle from Addis Ababa University, and also Professor Ndalkacho Sahaladamse from the United States EPA. So in this short presentation, I will try to touch core points from the thesis, such as background of the study, statement of the problem, hypothesis, significance of the work, and objective. Then after, I categorize the method and result part into 3 based on the project component. Finally, we will see the conclusions drawn and future work recommended. As you all know that water is associated with every socio-economic development. So in one or in another way, water availability is directly associated with the overall sustainable development goal. However, the world is now facing high water scarcity, either due to economic shortage or physical stress. Currently, around 40% of the global population faces severe water scarcity, and this figure is even expected to increase to 60% by the coming 2025. So as a result of the high water demand, now the water withdrawal it's outpacing the supply. If we see, for instance, this water stress graph, which is predicted for 2014, those regions highlighted with red and brown indicates a withdrawal to supply ratio of more than 40%. We can mention different factors that cause this water stress, but it is mainly associated with the rapid population growth, which is obviously accompanied by high agricultural and industrial activities. And these 2 sectors, they not only consume a huge amount of water, but they may also pollute the existing freshwater resource if the waste is not treated properly. Moreover, climate change is also exacerbating the water stress by making the water distribution uneven and unpredictable. So in order to tackle this major challenge, different solutions have been proposed and practiced so far. For regions, especially with economic shortage, we can increase the existing water supply through this water distribution system and the overall infrastructure. But in order to increase the water supply, either we can treat the water and reuse, or the other viable option is through desalination. And this desalination is not a new technology, because in this process, it considers the use of the vast brackish and saline water, which 97% of the world water is contained. So currently around 100 million metric cube of water is produced through these desalination processes. However, the conventional desalination technologies, they are challenged by both technical and economic barrier. Both thermal or membrane-based desalination technologies, they are still considered as energy intensive, and also a high initial investment cost is required to build those big sophisticated desalination plants. Moreover, it also has a negative environmental impact due to greenhouse gas emission and brine discharge. As a result of this, scientists are still looking for an alternative desalination method which can operate with low energy and minimal environmental impact. As you know that the world is now shifting to this biological desalination treatments. As a result of this, biodesalination became one of the emerging technology as an alternative energy, desalination process. Microalgae-based desalination is one type of biodesalination that utilizes microalgae to remove salt from either seawater or brackish water. One of the critical steps in this technology is selecting suitable algal species appropriate for desalination. So one of the major criteria is the algae that we select needs to be both salt tolerant, and it must have a good potential of salt removal. We can get such kind of an algae either by screening from a different algae collection bank, or we can directly screen microalgae from a saline habitat. So after getting the appropriate salt-tolerant algae, the next step is cultivating the algae in the water to be desalted. So in this case, we need to supply a certain nutrient in order to enhance the algal growth. Since microalgae are photosynthetic microorganisms, by fixing carbon dioxide from the atmosphere and also using sun radiation as an energy source, they do their own food through photosynthesis. So through this natural photosynthetic process, salt is believed to remove both through bioabsorption and bioaccumulation. Therefore, once this treatment has been done, the next step is separating the biomass from the water treated, so that the water that we get after the treatment is believed to have low salinity as compared to the feed water, so that we can use it for different purposes where high quality of water is not required. The second option is we can use algae-based desalination as a pre-treatment for other conventional desalination technologies, such as reverse osmosis, so that the overall desalination process cost will be minimized. In addition to this desalination, or in addition to changing the saline water into fresh water, this algae-based desalination also do have an added advantage, which is the production of green energy, such as biodiesel from the biomass harvested. As I said earlier, the problem is there is a growing demand for water, either for domestic or agricultural or industrial use. However, the available of fresh water on Earth is very limited, and on the other hand, a huge amount of water, which is saline water, is contained in oceans and seas. And desalination technology is a viable option which increases the water supply beyond the hydrological cycle. However, it is challenged by technical and financial barriers. On the other hand, untapped potential of microorganisms for desalination is not studied very well. And studies on microalgae-based desalination is at the very early stage of research, and additional investigations are required in order to mature this technology so that it will be applicable in the future. So the hypothesis that we set as a null hypothesis for this research is microalgae originated from a saline lake such as Lake Basaka have no potential to desal saline water, and also the biomass which is harvested at the end of the treatment is not suitable for biodiesel production. So the alternative hypothesis will be the opposite of the null hypothesis. So as I mentioned earlier, this algae-based desalination is still at the very early stage, and it's very far from data saturation. Therefore, the output result from this project will be helpful as a baseline data, information and knowledge for similar investigations in the future. Moreover, the output will improve the progress of this microalgae-based desalination, which is integrated with biodiesel production to produce both water and energy. So through a long term, when this desalination becomes matured and applicable, this integrated system addresses the water scarcity, the energy demand through climate change management. Therefore, this integrated system directly touches for the implication of sustainable development goals 6, 7, and 30. However, as I said earlier, water is crucial in order to meet the overall sustainable goals. So as a general objective, in this project, we evaluate the potential of microalgae, which are isolated from Lake Basaka, both for simultaneous desalination and biodiesel production. In order to achieve this, we set around 6 specific objectives that starts from salt-tolerant microalgal species screening and isolation from this lake. After that, we study the effect of light, temperature, and salinity on the algal growth and biomass production. Then after, we examine the desalination potential of this microalgae at different salt levels. We also assess the effect of salt on the microalgal lipid content, and we convert the lipid extracted into biodiesel, and we characterize the quality of the biodiesel based on the fatty acid methyl ester profile. So the first component of this project is screening and isolating salt-tolerant algal species from the algal assemblage. So in order to do this, the first step is sample collection. So we collect a graph sample from 4 different locations around the lake, and we mix the sample in equal proportion to get a composite sample. We fix 250 ml of the composite sample in glucose iodine solution, and we enrich 100 ml of the composite sample with Biodesalination medium to allow algal growth. Meanwhile, physicochemical characteristics such as pH, temperature, and conductivity of the lake water is measured at each sampling site. So after collecting the water sample, the next step is microalgal identification. So in this project, we identify the microalgae based on the morphology of the algae so that we identify it at the genus level. Throughout this experiment, we use an artificial growth media, which is a bolus basal media, and to prepare the agar plate, we use 1.5 weight by volume percentage of agar powder. So every media or any lab equipment is autoclaved prior to use. In order to grow the microalgae under controlled condition, we obviously need a growth chamber. Since we couldn't get a growth chamber both on the market and in our lab, we just construct locally such kind of a wooden growth chamber, which do have a fluorescent tube to adjust the light intensity, and the fluorescent tubes are attached to a digital timer. So for all experiments, we fix 12-hour dark and light photo period. In order to control the temperature here, we put 2 big water bodies, and air is provided from the atmosphere to the chamber through inverted small fans that are fixed on the chamber. The ambient temperature inside the growth box is measured based on a digital thermometer, which is fixed here on the chamber. So once we grow the overall microalgae sample from Lake Basaka, the next step is screening the most salt tolerant one from the algal assemblage. In order to do this, we subculture the stock inocula into bolus basal medium containing from 0 to 10 grams per liter sodium chloride. So we regularly investigate the different algal species at each salinity level, and we identify the most abundant species that grows well at the highest salt level of 10 grams per liter sodium chloride. So after identifying the salt-tolerant algal species, the next step is isolating unialgal salt-tolerant species from the algal assemblage. In order to do that, we follow standard serial dilution and plating method. Now let's see the results that are achieved from the first part. So this table shows some physicochemical parameters of the lake. As you can see from the electrical conductivity and TDS measurement, Lake Basaka is categorized as a brackish lake, and also the pH is in the alkaline range. The temperature which is recorded in the morning it's above 26 degrees Celsius, but previous studies record a temperature as high as 35 degrees Celsius. This could be associated with the volcanic activity of the Rift Valley. In the preliminary assessment of this microalgal diversity in the lake, we have identified around 19 species, and we subgroup them as green microalgae, cyanobacteria, and diarrheans. And as you see from the table, common freshwater green microalgae such as chlorella and synedesmus also observed. We also observe different cyanobacteria such as Arthrospira, which do have a great biotechnological application, especially in food industries. We observe also different freshwater and marine diatoms like Cyclotella, Neschia, and Navicola, and others. From this species screening, we have selected Synedesmus species for further investigation or for our study, because we found that this species is abundant both in bolus basal medium containing salt and also in bolus basal medium without additional salt. Previous studies also demonstrate that even though this Synedesmus species is a freshwater green microalga, it is found as salt tolerant, and also it is a good candidate for biodesalination application. And previous studies also shows that the lipid content of algal biomass from this species, it is more than 20%, so that we can use it for biodiesel production. So from the first project component, what we got is we prepare a uni-algal semidesmous stock solution for further study. In the second component of this project, we study the effect of culture conditions such as light, temperature, and salinity on algal growth and biomass production. In order to study the effect of temperature and light level, we put a 5-day-old inocula, which is 10% volume by volume, into boldest basal medium, and we allow it to grow for 16 days in the growth chamber. Since we studied the combined effect of temperature and light level, we use a full factorial design. That means for each combination of light and temperature, we did a separate experiment, and in total, we have done 12 experiments, and for each experiment, we did a triplicate analysis. So from the first experiment, we can identify the best temperature and light level for algal growth. So after identifying this best temperature and light level, we study the effect of salt on algal growth at the identified temperature and light intensity. To study the effect of salt on algal growth, we prepare a boldest basal medium containing from 0 to 15 grams per liter sodium chloride. So just like the previous experiment, we used 10% of 5-day-old culture. But in this case, instead of adding the suspension into the media, we centrifuge and wash the pellet, and we use that pellet as an inocula. So we allow the culture to grow in the growth chamber for 20 days. In order to monitor the algal growth, we use two methods. The first one is the algal cell density. That means counting the number of cells in ml of culture. So this algal density will be changed into specific growth rate and doubling time using this very known equation. The second method is the dry cell weight measurement. So at the end of the batch culture, we took 50 ml of culture. So we filtered it and washed the pellet, and we measured the weight of the pellet after oven dried. So we can calculate the dry weight biomass in milligram per liter using this equation. Throughout this research, we use one-way analysis of variance for data analysis, and to compare the difference between each treatment, we use two case studies, and the probability less than 5% is considered as significant difference between treatments. Now let's see the results on the algal growth profile. This is the XY graph showing the effect of temperature and light level on the specific growth rate. As you can observe from this graph, the specific growth rate increases as we increase the light intensity from 20 micromole of photon to 40, then to 65 at all temperature levels, except at 35 degrees Celsius, which shows an increase in specific growth rate at 40 micromole of photon. However, this change is not significant. Similarly, as the temperature increased from 20 to 25, the specific growth rate also found to increase. But what we observe is the effect of light is by far significant than the effect of temperature on algal growth, because as we increase the temperature from 20 to 25, we observe no significant difference in the specific growth rates. However, when we further increase the temperature to 30 degrees Celsius, the specific growth rate increased significantly. However, further increase in temperature to 35 degrees Celsius, results a decline on the specific growth rate. Similarly, the dry weight biomass also confirms this result. And as you can see here, for example, at each temperature level, as the light intensity increased from 20 to 40, then to 65, the dry weight biomass also increased significantly at all temperature levels. And also, as the temperature increased from 20 to 30 degrees Celsius, the dry weight biomass also increases. However, further increase the temperature to 35 degrees Celsius leads to a reduction in dry weight biomass production. So from this graph, we can clearly see that a temperature of 30 degrees Celsius and a light level of 65 micromole of photon, it is found the best light and temperature level for the microalgae selected. Now let's see the effect of salt on algal growth. As I stated earlier, the effect of salt is studied under this identified temperature and light level. So this graph shows effect of salt added on in the media, and the line here shows the change in specific growth rates with salinity, and the bar graph shows the change in dry cell weight at different salinity. As you can observe from this line graph, the specific growth rate at 2.5 and 5 grams per liter sodium chloride is found higher than the control, even though this change is not significant. However, when we further increase the amount of salt to 7.5 grams per liter, the specific growth rate starts to decline again. Similarly, the dry cell weight at 2.5 and 5 grams per liter sodium chloride is found higher than the control. However, cultures treated above 5 grams per liter sodium chloride, they observe a lower dry weight biomass. So from this growth profile, we can say that Senidesmus species, which is isolated from Lake Basaka, it can tolerate a wide range of temperature, light intensity, and salinity. However, the optimal growth condition was obtained at a temperature of 30 degrees Celsius and at a light level of 65 micromole. And this value is comparable with previous studies done on different Senidesmus microalgal strain. And the algae also can tolerate a sodium chloride concentration as high as 15 grams per liter concentration, and a medium salt level of 2.5 grams per liter and 5 grams per liter sodium chloride found to enhance the algal growth, and the maximum specific growth rate of 0.231 per day is observed in the presence of 2.5 grams per liter sodium chloride. And moreover, the dry weight biomass obtained from culture treated with 2.5 and 5 grams per liter sodium chloride is around 450 milligrams per liter, which is higher than the control. Now let's come to the last part of this project, which is evaluation of the desalination potential of Senidesmus species and also the lipid content at different salt levels. And finally, the quality of the biodiesel produced from algal lipid. In order to evaluate the desalination potential of this microalgae, we use electrical conductivity measurement. So in order to do this, we took 30 ml of culture and we filtered it, and we measured the electrical conductivity of the supernatant every 4 days interval, so that from the initial and the final electrical conductivity measurement, we calculate the desalination rates in percentage. But in order to understand how much total salinity is removed from 1 liter of saline media, we convert this electrical conductivity measurement into salinity in gram per liter. Even though this conversion it's a rough conversion using mathematical equation, it helps to understand how much salinity is removed from a saline medium. So at the end of the desalination experiment, the next step is lipid extraction from the biomass. So we harvest the biomass grown at different salinity level, and we wash the biomass and dry it and ground it into smaller powder, and lipid is extracted from this grounded algal biomass using a slightly modified Blein-Dyer method. So this is an old lipid extraction method, which is a solvent-solvent extraction using chloroform and methanol as a solvent. So we extract the lipid using this solvent, and finally we measure the weight of the dried lipid produced. So from the ratio of weight of lipid with that of weight of the algal powder used, we calculate the lipid content, the percentage lipid content. And this percentage lipid content is converted into lipid production by multiplying it with dry weight biomass that we calculated from during gross examination. So the final step is biodiesel production and characterization. As you know that oil or lipid can be converted into biodiesel using the transesterification process. So for this transesterification process, we use methanol as a solvent and potassium hydroxide as a catalyst, and oil to methanol ratio is 1:11, which is based on previous study done here in Addis Ababa University, and the experiment is carried out at 60 degrees Celsius. So the fatty acid methyl ester, that means the biodiesel, is analyzed using GCMS, and this experiment is done at the organic laboratory here in Addis Ababa University, and I would like to thank Dr. Stefanos for his cooperation. So when we come to the result part of this project components tree, as you can see from this XY graph, which shows the change in electrical conductivity with cultivation time, the electrical conductivity, it increases gradually, and the maximum electrical conductivity reduction occur at the end of the batch culture for all treatments except for the control, at which the maximum electrical conductivity reduction at the 16th day, and for the culture treated with 5 grams per liter sodium chloride, the maximum electrical conductivity reduction occurred at the 12th day. So this is a bar graph showing the relationship of salinity with maximum desalination efficiency. And as you observe from here, we didn't get a direct relationship between desalination efficiency and salt added, because the desalination efficiency for the control is around 21%. So it increased to 23% as the amount of salt added increased to 2.5 grams per liter sodium chloride. Then it starts to decline until the addition of 10 grams per liter sodium chloride, and it started again. So we didn't get a direct relationship. However, from this graph, we can observe that the maximum desalination efficiency of around 20% was obtained for the culture treated with 2.5 grams per liter sodium chloride, and the least, which is 15%, was obtained for the culture treated with 10 grams per liter sodium chloride. But when we consider total salinity removal, as the amount of salt added increased from 0 to 15 grams per liter sodium chloride, the total salinity removal also increases. Therefore, sometimes 23% electrical conductivity reduction from culture treated with 2.5 grams per liter sodium chloride is less than removing 15% from a culture containing 15 grams per liter sodium chloride, and the maximum salinity removal of around 3.13 grams per liter was achieved from the culture treated with 15 grams per liter sodium chloride. So now let's compare what we got with previous studies. As you can see here, the previous studies performed on examining the desalination efficiency of different synidesmus strain, and different study used different operating conditions such as media used and light level temperature. If we see the salinity range, which is tested before, a maximum of salinity, which is 10 grams per liter, is tested in figure study. But the salinity range tested in our study it's a bit wider as compared to previous studies than only on the synidesmus species. When we compare the desalination efficiency here, around 30% was reported from Sahala Damiste study at a salinity of 4 grams per liter. And as you can see here, there is also 26% from Gan et al. Study, which is done in 2016. Figler also reports a range of desalination from 3 to 26% in the salinity range of from 0.5 to 10 grams per liter. So the result that we get from the present study is its best fits with Figler study, but still it is within the range of previous studies done on different synidesmus strains. When we come to the effect of salt on lipid content, as you know that the algal lipid content found to enhance under stressed conditions such as nutrient starvation and higher salinity. And the lipid content of the control is around 20%, which is comparable with previous studies done here in Addis Ababa University. And as the amount of salt added increased from 0 to 10 grams per liter sodium chloride, the lipid content, which is the bar graph here, also found to increase. And a maximum lipid content of around 26% was obtained from culture treated with 10 grams per liter sodium chloride. This is 27% higher than the control. However, due to a reduced in algal growth as higher salinity, the maximum lipid production is observed from the culture treated with 5 grams per liter sodium chloride, which is around 109 milligrams per liter of lipid. So when we summarize the overall batch culture at different salinity, as you can see here, synidesmus species isolated from this lake can be used for the simultaneous desalination and lipid production process. However, we found that culture treated with 5 grams per liter sodium chloride, it best suits for this integration system or in this integration process of desalination and lipid production. Therefore, what we did is we convert the lipid, which is extracted from this culture, into biodiesel, and we characterize the biodiesel using GCMS. This is the GCMS spectra. So we change this spectra into qualitative and quantitative data based on the peak area, retention time, and also other information from the GCMS library. So as you can see here, the percentage of saturated and monounsaturated fatty acid of this biodiesel is around 69%, and biodiesel containing higher proportion of saturated and monounsaturated fatty acid is considered as good quality biodiesel. However, a certain portion of polyunsaturated fatty acid is also important, and in our case, the remaining 31% accounts for polyunsaturated fatty acid. When we see in detail the different fatty acid methyl ester, here we do have a fatty acid methyl ester from carbon chain 16 and carbon chain 18. And chain length between carbon 16 to carbon 18 is considered as a good characteristics for quality biodiesel. Moreover, the percentage of polyunsaturated fatty acid, that means fatty acid with 4 or more than 4 double bond must be less than 1% according to the European Union biodiesel standard. In our case, we don't have such kind of polyunsaturated fatty acid. This is also a good indication about the quality of the biodiesel produced. So the only value which is out of the standard is the concentration, I mean the composition of linolenic acid, which is a maximum of around 12% is allowed. But in our case, we get around 20% of linolenic acid. Even this value, 20%, is lower than previous studies done on Synidesmus species grown under saline condition. However, in order to improve the overall quality of the biodiesel produced, we can mix this biodiesel with other biodiesels having lower linolenic acid so that we can blend it. So as a general conclusion, we can say that Lake Beseka it's a saline alkaline lake, which is rich in diverse microalgal species with great biotechnological application. This species can tolerate a wide range of temperature, light level, and salinity. However, a temperature of 30 degrees Celsius and a light level of 65 micromole of photon is the best condition. This species can grow in the presence of salt level as high as 15 grams per liter sodium chloride. However, a salt level of 2.5 and 5 grams per liter sodium chloride found to enhance the algal growth. In addition to salt tolerance, these microalgae also have a good desalination potential, and a maximum desalination efficiency of 23% was achieved from culture treated with 2.5 grams per liter sodium chloride. Moreover, the biomass harvested after the treatment could be converted into lipid, and this lipid content is found to enhance under salt stress. And for culture containing 5 grams per liter sodium chloride, as I said earlier, we found this is the best salinity for this coupling system. And at this salt level, a desalination efficiency of 17% and lipid production of around 100 milligrams per liter was achieved. Moreover, the biodiesel, which is produced from this lipid, it do have a good characteristics for future use. So the overall result from this project disprove our initial null hypothesis that salt-tolerant algal species originated from this saline lake, it could be employed for simultaneous biodesalination and biodiesel production. So before finishing this presentation, I will just try to put some pointers for future insight as a gap. As I said earlier, even though this alternative desalination technology, it seems fascinating, but still the research is at its very early stage, and research focused on exploring the potential of different algal species is required. Moreover, studies at the molecular level focused on identifying the gene which is responsible both for salt tolerance and desalination needs to be investigated. Since most of the research done so far are at the laboratory scale, except few studies, pilot scale conditions under natural environment also needs to be done. And one of the problem in converting this salt-grown algae into some product is a pollutant transfer, in this case salt, because when we convert this algal biodiesel grown in saline medium into bioenergy, the salt which is laden inside the cell may be transferred to the final product, and the effect of this needs to be investigated. Last but not least is the bottleneck for any biotechnological application involving microalgae is algal harvesting from the biomass, which is a very energy intensive and costly. Therefore, in order to minimize this, the use of fixed bed and biomass cultivation for this biodesalination application also needs further attention. So when we come to this publication, so far we have published 2 papers on peer-reviewed journals, and also part of this work is presented on the American Chemical Society annual meeting on 2021, and the abstract of this presentation also published on Science Direct. Moreover, a poster presentation was made in different sessions, in different scientific sessions prepared by Addis Ababa University. Before finishing this presentation, I would like to express my deepest gratitude to my advisors, Professor Kalek Azauga, Dr. Demek Akafli, and Professor Ndalkacho Usala Damse, for their unlimited support, starting from proposal development until thesis writing and publication. I also would like to thank Jimma University for letting me study this PhD as a full-timer, and also the African Center of Excellence for launching such an incredible original program that we not only share knowledge, but we also shared culture with different students from different parts of Africa. Finally, I would like to thank the Ethiopian Forest and Research Institute, because all the experiment is done in this institute. They provide me space, instrument, and everything for not less than 2 years. Finally, thank you very much.